Hi, everybody, and welcome back to our seminar series for the new semester. Hopefully you should have all, if you're on one of the CMDI mailing lists, you should have gotten a calendar invite to add a recurring event. You might have gotten a couple actually, to add this event to your calendar so you never forget. 3 o'clock on Fridays are our microbial dynamics seminars. So for our first week back here, we are going to have a practice job talk from James Gurney in the Brown Lab. And he's going to talk about his research looking at how to improve phage therapy in a informed way. So with that, I'll kick it over to you, James. Thank you.
Thank you. Yeah, as Jennifer said, I'm a postdoc in Sam Brown's lab. I've been invited to submit a faculty package for University of Tennessee and present a seminar series. So hopefully, this is geared at presenting to a department. Happy for any criticism, constructive or otherwise, at the end of this. But yeah, let's get started. So I've been working on phage therapy for the last 5 or so years now, and I'm interested in how we can use evolution to improve strategies for treating bacterial infections. So what's the big picture here? We're talking about antimicrobial resistance and chronic infections. So the figure down here on the left, let me turn my laser pointer on before I forget. The figure down here on the right is the sort of the timeline of antimicrobial resistance since the 1930s. Phage actually predate this a few decades earlier. But every time we've discovered a new antibiotic, more or less a decade or so later, we have discovered a resistant strain to that antibiotic. And there was, this is called the dark ages of antimicrobial discovery from the 70s to the early 2000s without much being really discovered novel. But we have time and time again, every time an antibiotic is discovered or designed or made, resistance emerges quite soon afterwards. It was pointed out to me recently that actually the majority of people who die of bacterial infections, they're not dying from antimicrobial resistant diseases, they're dying because of a lack of antibiotic. So there's two faces of the story. There's one, we need to improve who people need antibiotics worldwide, but we also need to think about how we use those and reduce resistance. That being said, again, it's often described as a problem for the future. Antimicrobial resistance is some monster on the horizon that we're going to have to tackle, much like climate change. It's happening later. Well, in reality, it's happening like climate change. It's happening today. There are people in the US who will die because of an antimicrobial resistant infection. So I'm particularly interested in chronic infections. These can be anywhere from a month to a decade to lifelong bacterial infections. I'm specifically interested in Pseudomonas aeruginosa infecting the CF lung, as we'll see later. But these infections can also be polymicrobial. They often are polymicrobial. They require long-term antibiotic usage. And due to shifting demographics and populations, we expect to see more people with more chronic infections. So a number of solutions have been put forward. I don't think any of these are a catch-all, magic bullet. But we definitely have to think about them in groups. So, for example, surveillance and stewardship, how we use antibiotics, where we use them, why we use them. Fantastic initiatives being put in place to try and reduce the exposure of antibiotics, especially in like agriculture, or not prescribing antibiotics for common colds. There's also a question about economics. Are we doing enough economically to incentivize pharmaceutical companies and other places, research, for example, us, academics, to spend time developing new antimicrobials? If you make a new antibiotic, you get a paper, amazing paper, no doubt, but also that antibiotic will then sit on a shelf until it's the last course for use. It's not going to get used. And a pharmaceutical company has spent hundreds of millions of dollars making this antibiotic just to have it sit on a shelf until its patent expires, useless to them. Unfortunately, as much as I think a lot of us would like to live in a more egalitarian world, that's where we find ourselves economically right now. So I'm interested in novel therapeutics, specifically phage therapy. So I'm going to talk about that. My phage, all this work is using is OMK01. It infects Pseudomonas aeruginosa. It targets an efflux pump. So if you're not aware, efflux pumps are one of the ways that bacteria can have antimicrobial resistance. So they pump antibiotics back out of the cell effectively. They're not just antibiotic pumps. They do other things. They've evolved for other reasons. There's other reasons bacteria have them. They do other functions. But for this case, in this scenario, we're just thinking about antimicrobial resistance pumps. Okay. So this phage binds part of this pump, probably the OPMR. We've sort of determined that from transposon screening. So one of the ways the bacteria can develop resistance to this phage OMK01 is to mutate this pump. So by changing this pump, it loses the ability, the phage loses the ability to bind that pump and infect the bacteria. This puts the bacteria in a catch-22 because obviously if you change the pump, there's a very good probability they also change its function of pumping out antibiotics. So the catch-22 is you develop resistance to the phage or you maintain resistance to the antibiotic. So we know that this system works in vitro. Here's a little bit of data. I'll show you more of this later. If you expose bacteria to this phage and then see who survives, take the phage away, see who survives, the surviving bacteria typically have lower antibiotic resistance than the initial ancestor bacteria. So my question in this case, is this evolutionary stable? Does it work over a treatment window? And how can we improve it? So let's start with an actual treatment. This is all, a lot of the work I'm showing today was done before what I'm about to show you. This is sort of, not to bury the lead because this is the really cool stuff. Colleagues up in Yale by Paul Turner and Ben Chan have been doing compassionate care release studies in CF patients. I'm charged with studying a lot of this Coevolution dynamics for a CF fellowship in R21, which I'll get to right at the end. But the story of this patient is a 22-year-old female patient with CF. She has had a pretty decent FEV1 score. So if you're not aware, FEV1 is how much volume of air you can force out of your lungs over a given time. It's a very good indicator of how physically fit you are. CF patients typically have stable states and they'll decline, stable and decline, eventually leading to either requiring a lung transplant or, unfortunately, mortality. She had a particularly stable FEV1 up until her until early 20s. And then she had what I think is quite common for a lot of CF patients. She was treated with multiple antibiotics. So in this case, IV colistin. You can see how the FEV starts to drop. They give IV colistin. It improves. It goes back down again. They switch from, they increase IV colistin to include 2 other antibiotics. It continues going down. She also develops what, again, not an uncommon condition. She has a C. diff infection because she's been on IV antibiotics for so long. The C. diff has been allowed to basically expand into that niche and take over and causes a very horrible disease. Fortunately, not fatal in this case, but it can be. She then enters an area of stability again for a few months. Again, this is the course of almost a year. Her FEV1 is bouncing up and down. And then she has a precipitate drop down. This is an exacerbation. This happens in CF patients where they'll have basically, we're still not sure what happens, but they will have a very quick decline in FEV and typically is associated with some micro changes, but not greatly. But the main case is that she has a very quick drop down in FEV1. Basically, she feels crappy. She gets very sick very quickly. Continues going down even after changing onto a new antibiotic, piperacillin tazobactam, which doctors don't like to typically give. This is an indication this is a serious case. They typically don't. They give it, but it's not common for CF patients to receive piperacillin tazobactam. Also, I'm told by clinicians, but in this case they did. And then they performed some clinical microbiology on the samples of the Pseudomonas pulled out here, and they found what I would consider basically the worst case scenario. The Pseudomonas taken out when screened against this long laundry list of antibiotics was completely resistant to all these antibiotics. These are the choices for therapy, and it's resistant to everything. So this is a patient who has entered what was considered a very risky point. There is no available treatment. Their FEV1 is still continuing to go down. In a lot of cases, this would lead to either death or severe morbidity. Fortunately, she was allowed to enroll in a compassionate care study using phage therapy. So we're zooming in here a little bit, but I won't keep you in suspense. For 20 days, phage therapy was administered and tracked. Her FEV1 bounced back up from mid 60s to mid 75, and she feels much better. I actually got to meet her at the CF conference in Nashville about a year ago. And having, a lot of my work is based upon stuff that happened in her lungs. It was very emotional me to actually meet this person who is potentially alive today because of the phage therapy. I always look at this and say, why can't we give it to more people? Why are we not doing this for more people? We have been. Paul and Ben have been, started a clinical trial. There's been more compassionate release cases after this. I'll draw your attention to the left hand here of this column again. So after the yellow and the green, pulling out more Pseudomonas aeruginosa, the gram here has changed from completely resistant to mostly susceptible or intermediate, and then right at the end, we're seeing the bacteria basically becoming highly susceptible to the antibiotics. This means that this patient has, had a successful phage therapy. Their FEV1 has responded. They were treated with antibiotics during this time as well, allowing then at the end to potentially reuse an antibiotic. So we could go and say, we've applied a phage therapy, it's worked, and if she gets sick again, she can take an antibiotic again, or, as I'll talk about later, maybe a phage again. So my two main research topics, which sort of address this sort of clinical aspect we've been talking about, are this thing I've called phage steering. This is the idea that I and lots of other people had the same idea. Phage impart selection on their hosts. Can we use that selection to steer pathogenic phenotypes in our favor? So reducing pathogenic phenotypes like antibiotic resistance and virulence, and maybe even transmission. So the second topic is phage coevolution and reusability. How can we build a better understanding of how phage co-evolve with their bacterial hosts and improve treatment robustness? And I'll get to that at the second half of this talk. So when we consider how conventional therapies fail, this doesn't have to happen in one patient. This can happen in a meta population, but I've drawn it as well. I've stolen diagrams to show it from one patient. You have a sick person, you give them antibiotic, they may get better. Sometimes the bacteria evolves resistance. You keep giving the antibiotic, the patient gets sicker and sicker. Eventually you end up at a point where you can just throw antibiotics to the bacteria and it has no effect and the patient dies, or again, it's very, very sick for a very long time. What about phage therapy? Are we going to end up in the same position? If we have, we typically start with a sick patient because of compassionate care. Maybe we shouldn't. I would argue, there's an argument to be made. We could treat less important, less deadly infections, as should say, with phage therapy as a front line, so we reduce how much antibiotics we use when they're serious, because they are. I still think antibiotics are the best drugs we have. They're the best things we have because they work so well. But perhaps we could take a step back and say, okay, well, you've got a sore throat. It could be common cold, or it could be a bacterial strep infection. Let's give you a phage. Maybe it self-resolves. We don't throw antibiotics at potentially a self-resolving infection. But let's look at phage therapy and why it might fail. Again, you give the phage to the person, they get slightly better. The phage are killing the bacteria. But are we then just evolving phage-resistant bacteria and the patient gets sick, and now you've let's say you're a drug company, you've spent hundreds of millions of dollars developing this new novel phage therapeutic, and it's useless after the first application for a patient. No, there's no pharmaceutical company that would want to touch that or make that or spend the money on that. Phage steering makes use of this idea that we're developing resistance to the bacterial populations. So I've schematized it here as this is the type 4 pili. The phage, we have phage that do this. I'll show you some data in a second. Phage uses as an attachment receptor to initiate infection. And of course, this isn't a phage receptor. The bacteria is using this for its own purposes, but the phage uses it as a way to attach to the bacterial surface. Bacteria can mutate these receptors, and that will change the virulence profile generated from that phenotype. For example, when we did this in a wax moth model, if we take the wild-type bacteria and ask how well it can kill the simple model versus a bacteria treated with this phage that binds that type 4 pili receptor, we find that the bacteria that have evolved resistance to that phage have a lower virulence profile. This case here, there are no phage present in this treatment. This is the bacteria after selection from the phage. So I've taken the phage out. Same thing here. You saw this data a minute ago. This is the MIC profile after being exposed to that phage OMK01. We have reduced MICs of the evolved bacteria. So how is this different from therapy? Just to make this concrete. On the left, we have the therapeutic data. So again, I'll explain this a bit more later, but we're basically looking at survival probability. Therapy is the idea that you give a phage to a patient, they get better. The bacteria decline in density. The immune system comes and mops everything up and acts like an antibiotic. Same as conventional drugs. Phage steering is this idea that you can apply selection and the bacteria will have a different phenotype afterwards. So again, in this point here, I think the easiest way to sort of clarify these two ideas, phage are present here on the left for therapy. Phage have been present, but can be taken away, and now you've changed the bacterial phenotype. So let's look at this case study. Is phage steering stable? This is some work I published last year. I did a coevolution experiment with phage and antibiotics. I used 3 different antibiotics, erythromycin, ciprofloxacin, and tetracycline. I evolved them for 20 days. Every second day I made a new passage, so there were 10 generations, right? So the simple question was, okay, well, no one had done this at this point. Has the phage still driven the antimicrobial resistance down over 20 days? Remember, this is its own KO1 phage. It binds efflux pumps. And what happens if you add the antibiotic? Like, does the antibiotic override the phage's ability to do this or not? So again, not to keep you in suspense, no is the answer. In the presence of the antibiotic, by itself, the bacteria develops resistance to the antibiotic. Not surprising at all. That's exactly what we hoped would happen. And of course, the phage, as we've seen before, reduced the MIC for these 3 antibiotics. So after 20 days of co-evolving with the phage, the bacteria had a lower MIC. The thing I find really interesting and really cool is that when you added the antibiotic and the phage, you were either as good as the control or better. So in this case, the tetracycline and cipro, the bacteria's MIC reduced in the presence of both the phage and the antibiotic for 20 days. So the phage was able to steer the bacterial phenotype towards less antibiotic resistance, even when the antibiotic was present. Then I moved on to do some animal work. My animal work at that time of choice, the animal model was this guy, the Galeria wax moths. These are fantastic for a number of reasons. One, they're very, very cheap. They're very easy to work with. They fit in 24 well trays. And they also die at the drop of a hat. So if you inject them with 50 to 500 CFUs of Pseudomonas, they will die within 48 hours. So anything you do to improve survivability is really obvious. So let's look at some of this data now. So this is a survival probability over 48 hours of a mock phage therapy in these animals. So the green circles here are the bacteria control, so just adding bacteria. I've left off the controls of like the vehicle control and the phage vehicle control by itself. They're 100% survivability. The bacteria kill, unsurprisingly, as I said, within 24 to 48 hours, almost everything is dead. The antibiotic had a slight benefit and improved survivability a little bit. This was using a sub-MIC concentration, so I was not expecting great survivability with the antibiotic. I'm using erythromycin here if you're interested. The phage, again, this is the therapy. It worked. The phage increased the survivability of these wax moths. We had about 50% survivability compared to 0 for just the bacteria. And the phage antibiotic treatment combined had a slight increase in effect. There's actually, the interaction here isn't significant, but again, there's a slight increase in effectiveness of survivability if you had the phage and the antibiotic. That wasn't what I was interested in, because we know phage therapy works. It's been done for 100 years now. We know phage will kill bacteria. We know it might work. The question I had was, have we steered the resistant phenotype in vivo? And we had. So me and a talented undergrad, she ground up a bunch of these wax moths and plated them out and took a bunch of colonies. And what we found was the antibiotic was still, in the presence of the antibiotic, the phage was still able to steer the infection. So in vivo, we can see a reduction in MIC even when the antibiotic is present. So this opens up this idea of non-inferiority treatment. So you can continue giving antibiotics, you can continue the same therapy, and still have this effect that we're after. Of course, there's some issues with this data. These wax moths are probably one of the most simple animals you could use. When you inject them, you have to inject them with the phage at the same time. So there's no time for the bacteria to develop an infection. I've done other studies with ex vivo pig lungs where we see a very similar effect, where we put the pig lung tissue in, we allow our bacteria to colonize it, and then we treat it 48 hours afterwards. We see the same effect, but there's no immune system there. Okay, so that's the first project, sort of looking at this idea of reducing resistance or steering phenotypes in vivo. I think I've shown that in addition antibiotics combinations with antibiotics, you can still steer bacterial phenotypes, and we can potentially apply this to clinical data as we've seen before. So on to the second topic. I'm going to talk a bit more about this idea of coevolution and reusability. So this is a nice little diagram from a video I helped write for Coastal's, and I think it again shows nicely this idea of there's a coevolution arms race happening, which I'll explain in a minute. So I'm interested in this idea of repeated dosing of phage. So again, if we have a patient who has received phage therapy, and in this case, and an antibiotic, can we then go back and give them the phage again? I don't think this idea is particularly crazy. As we saw with the patient, they received multiple doses of antibiotics through a year. We have to be able to think about how we reapply a therapeutic. That's just the case with chronic infections. Again, the CF is a lifelong condition. You're going to have to reapply therapies at some point, and it'd be nice if we could do it with the phage. If you're the FDA, you're going to approve a phage therapeutic, and I want to give it to someone, it has to be the same phage. It has to be the phage that they've approved. I can't just do a coevolution experiment, pull out phage at the end and say, well, these ones have evolved to counteract these ones. I have to be able to go back and use that phage. So let's just schematize this with the patient data again. So I've superimposed on a worst case scenario for her later on. Let's say some time goes past and the bacteria rebound. There's still some, they still found Pseudomonas in her lung. It didn't eradicate the Pseudomonas. There was still Pseudomonas there. So maybe a year, maybe 5 years, maybe 10 years, she has an outbreak of that bacteria again. The resistance is developed, etc., etc. So her FEV1 is tanking again, let's say. The question then becomes, well, what if we give phage therapy again? Can we reapply phage therapy? If it responds positively, and again, I mean reapply the same phage. If it responds positively, I would consider that a robust treatment. We've been able to re-administer that same phage. And again, that's the approved phage. That's the phage I have to use in the fridge. If it doesn't, I would consider that's a failure. I think we can think of this in terms of coevolution. So phage and their bacteria co-evolve. Again, so I did 20 days. That's how long the bacteria and phage were found to be inside the patient. We know we find coevolution dynamics happen over 20 days. And the sort of the main take-home question for this work is, can a standard phage work on an evolved bacteria? This is then now, as I said, it becomes a coevolutionary question. And the way I tend to think about coevolution is in these arms race versus fluctuating selection dynamics. A simple way to consider this is, are you developing generalized resistance or specialized resistance? And the way I like to talk about that is, imagine you're a gardener and you have a flower in your garden you want to protect, and there's an intruder who wants to get into your garden. So the first thing you do is you build a gate. Fine. The intruder can't get in. They then work out how to jump over the gate. You then build a wall. They then buy a ladder. You then buy a stick that pushes the ladder off the wall. They then build a ladder that has a little flip thing that opens up when you try and push off the wall. That's an arms race. Every single step, the ladder right at the end will still work on every previous way to get into that garden. The way around that, there's a specialized resistance. Let's say you buy a gun, or something to change the system from just being a wall-based system. Now the intruder has to work around that new thing. So they've moved out of that arms race dynamic into something more specialized. Same thing happens with bacteria and phage. Lots of papers have been published on this idea of how bacteria and phage co-evolve, and how stress might affect it. Increasing stresses will probably shift from ARD to FSD. So what am I talking about with arms race dynamics? So there are two types. There's arms race dynamics, ARD, and FSD, fluctuating selection dynamics. These are technically on the same mathematical continuum. ARD on one side, FSD on the other side. So if you move away from ARD, you're by definition moving towards FSD. And again, a simple way to think about it is, ARD is kind of like an antibiotic, how bacteria develop resistance to antibiotics. They get better and better through time. In this case, we're looking at how well a bacteria resists a phage. So we take bacteria from time point 1, time point 5, and time point 10, and we ask how well can you resist the phage from the middle time point. So in T1 versus T5, that's basically the past bacteria fighting a future phage. So we expect under arms race dynamics to have a low resistance to that future phage. Again, going back to the garden analogy, this is the intruder with the nice tall ladder, and this is the gardener with just a fence. Doesn't stand a chance. We move through evolutionary time, and your resistance will increase to the point where you're now the bacteria in the future, the T10 bacteria versus the phage from T5. You now have a generalized resistance mechanism against a lot of things that come before. Again, as I said, anything else is FSD. That's the cheat sheet here. So we can do the same thing for phage infectivity. I've drawn it this way just so, again, from bottom left to top right, if it's going up like that, it's ARD. This chevron pattern here for FSD doesn't have to be that pattern. It can be something else. But if it's not ARD, it's FSD, okay? And as I said, the cheat sheet here, I'm not saying it's ARD, it's FSD. And we have other ways we have other FSDs. For example, we have FSD which can look like this, right? So we've lost that chevron pattern and we're going in a different direction. So in this case here, the past phage is actually better at killing the future bacteria, which sounds crazy, but it does happen. Like I'll show you some data in a minute. We can have that. So we can have FSD can lead to dynamics where we have phage from the past being able to kill bacteria better from the future. And as I said earlier, ARD will shift into FSD when costs increase. And things that will increase the costs that we know so far are changing the environment. For example, having low amounts of glucose available in the environment seems to shift it. Having predation from different animals like C. elegans seems to shift it. No one, as far as I'm aware yet, has studied how antibiotics shift it, so that's what I've done. And again, they seem to shift it. So increasing the cost or the stress of the environment will shift ARD into FSD. So again, if you think about our ladder analogy, you can't just keep building a taller and taller ladder. At some point, it becomes the cost of investment in the ladder becomes worth more than whatever you're trying to steal. So that will shift into a different mechanism. So what I did was I did another coevolution experiment. That's my go-to, is to do coevolution experiments. I used the OMK01 phage with 3 different antibiotics. This time I chose clinically relevant antibiotics because the first time I didn't. I'm also using SCFM2, which was developed by the Whiteley lab. This is a media which mimics the transcriptional profile for Pseudomonas inside CF sputum. So when the CF patients have this very thick sputum in their lungs, Marvin Whiteley's lab have developed this media which has very similar macronutrients and viscosity and all this other cool stuff. So as far as I'm concerned, this is a very, very close to actually inside without going into an actual patient's lung way of testing this. And I used antibiotics. So to test this sort of impact, how well bacteria resist, how well phage infect, I used a cross-streak experiment. So again, taking the bacteria from different time points, 1, 5, and 10, and I cross-streak them against the phage. So in this case, it's the phage from T5. So bacteria from time point 1, cross-streak T5 phage, most of the bacteria are killed. That's to be expected under ARD. Taking them at the same time points for T5 and T5 phage. There are a couple of sensitive ones down here. So again, if the bacteria don't grow across the entire way, it means the phage was able to kill it. But when we get to the end, basically all the bacteria are resistant to that phage. So this is showing ARD dynamics. So I did that for all these different isolates, picked out a whole bunch, and tested them. And what I found, when we just have the phage and the bacteria co-evolving, no antibiotics, we find what is almost perfect ARD dynamics, bottom left to top right. So bacteria from time point 1 were less resistant to phage from the future. Bacteria from the future were more resistant to phage from the past. Fantastic. ARD dynamics. This dashed line. Same thing for infectivity, dashed line. Now this is kind of to be expected. I would have thought the SCFM2 may have pushed them into ARD dynamics because it's a nice media, but it's not the nicest of medias. But I guess the bacteria didn't seem to mind. They had enough the ability to maintain these generalized resistance mechanisms and maintain ARD dynamics. That all changed when we added antibiotics. So when we added, in this case, I'm looking at Cipro. I have the data for the other 2 antibiotics, but we'll just focus on Cipro for this talk. the dynamic goes out the window. We lose that bottom left to top right response, and instead we enter into something which is just different. This is Fsd Dynamics. Well, potentially Fsd Dynamics. There are other ways to test this, but for now, this looks like Fsd Dynamics. The thing that really interests me here, as I said earlier, is we have I've marked with this little phi symbol. We have a phage from the past, which is as good or maybe slightly better than the phage from the future at killing a future bacteria. So this speaks to this idea of this is robustness. We could potentially give a phage from the past and treat an evolved bacteria. So that's exactly what I tested. I went and looked at how well the ancestor phage will kill these evolved bacteria. So what we're looking at here is the PO1 from the fridge. So this has never seen this phage. It's just, as a control, PO1. It has almost no resistance to this phage. If you put these two things together, the phage completely obliterates this bacteria. The thing marked phage here is these are the bacteria at the end of the experiment that evolved with the phage. There are no phage present in this treatment. This is asking how well they resist the phage. So these have co-evolved with the phage. This is the bacterial resistance to the ancestor phage. What we're seeing, again, completely understandable by ARD dynamics. The bacteria that evolved with just the phage is almost completely resistant to the ancestral phage. That all changes when we added the antibiotics. So for the other two, pip and toad, we saw difference, but not as big as cipro. So this speaks to this idea of robustness. This bacteria co-evolved with the phage and the antibiotic for 20 days, the same window of time as we saw in the patient, and at the end of it, the ancestor phage was still able to kill over 80% of all the bacteria tested in that population. That speaks to we don't know why this has happened, we don't know how it's changed, but we know we could potentially give this phage back if a patient exhibits this dynamic. So my working hypothesis, which we're working, we've got a grant to explore this now, is that if you think about coevolution and phage dynamics in therapy, I've schematized it here as red and blue, red for bacteria and blue for phage. This is sort of an evolutionary argument. So the phage kill the bacteria declines, and this isn't predator-prey. There's some evolution event that happens here which allows the bacteria to start increasing in frequency again. And same thing, the phage develops some sort of counter to that tit for tat and enters into a coevolution dynamic. My argument is if we were then to reapply the phage, we either have failure or reuse. So if at this endpoint here, the bacteria has maintained a generalized resistance, and then we try and reapply a therapy later, it will fail. However, in the presence of antibiotics, we might have pushed it into a different coevolution dynamic, so we can reapply a phage and potentially re-enter another coevolutionary dynamic and give it to the patient. So just to finish up, because this was that OMK01 phage that steers bacterial phenotypes, I of course did the MICs, and again we saw a very similar pattern to what we saw in the previous coevolution experiment. We see if you have phage and antibiotic present for 20 days, the phage is still able to steer the bacteria into a lower resistance profile, and that's what we saw in the patient, to be honest with you. Again, we see this, the phage and antibiotic were there, and we saw a reduction in MIC for these three antibiotics, the same three I've used here. So it's tobramycin, ciprofloxacin, piperacillin, and tazobactam. This phage works in patients. It's able to treat Pseudomonas, and it's able to steer, and potentially, if we understand the coevolution dynamics again, if this patient gets sick in the future, she may be able to get the same phage again if we can understand these outcomes. So I'll wrap up now by saying, hopefully I've convinced you of two things mainly: that phage are able to reduce the virulence and antibiotic profiles of bacteria. They can do this in vivo, they can do this in a therapeutic, and they can do this over a therapeutic window as well. And I've hopefully also convinced you that phage therapy potentially has a trick up the sleeve that other conventional therapies don't. It can potentially be made more robust by manipulating the coevolutionary dynamics and shifting them into something more favorable. And luckily, it seems the way to do that is to give them antibiotics, which is what we'd hope to do at any rate. So just to follow on, this work is following on now to a CF fellowship, which I just got with the explicit goal of testing these ideas in samples collected from CF patients who received compassionate release phage therapy. So we have 8 patient samples taken through time who received different phages, and I've got the ability to go back and now look at it and say, okay, how have they co-evolved? Has it increased robustness? What antibiotics were they taking? We also have 2 R21s on the topic. We had one funded for the last 2 years, and we just got another one funded this year, asking very similar questions, but luckily distinct, using the same clinical samples. We're going to investigate questions like, have phage been included in the CRISPR-Cas system in these CF patient isolates? An interesting thing is when we look at CRISPR-Cas in CF patients not receiving phage therapy, it doesn't seem to do anything. We don't see the endemic phage that are in the CF lung being taken up into that system. It's pretty static. No one, as far as I'm aware, has looked to say, well, what about phage therapy? Is CRISPR-Cas important for phage therapy? I get asked that quite a lot. Like, okay, well, you're assuming resistance is going to be developed by the envelope, but what about CRISPR-Cas? I have a paper on that if you're interested. But yeah, I'll hopefully be the first person to actually look at, do we see CRISPR-Cas uptake inside CF infections? I'm also working on generating the pilot data, asking questions about how can we block transmission using phage steering, which I'm hoping to be an independent award when I get my first faculty position. That leaves me with time to say thanks to the Brown Lab. I'm actually not in this pitch of 1,000 people in our lab at some point. And this work was started in Mike Hopper's lab in Montpellier. Of course, thank you to all the funders, specifically now the NIH and Cystic Fibrosis Foundation for giving me a fellowship, and of course the collaborators in Yale, Paul Turner, Ben Chan, and John Koff. And thanks to the team from COSAT for letting me steal their lovely artwork. And I'll leave it there.
Right. Thanks, James. That was awesome. So we'll open it up now to questions from the audience. So you can either just ask your question. Ask it for you.
I have a question if nobody else has got one right now.
Sure, go ahead, Steve.
So you showed like the case of the girl who had CF where they, you got the exacerbation, she got a reduction in FEV1, they treated with the phage and she got better, suggesting that the phage worked, suggesting that it killed Pseudomonas, I guess.
They did do data to show that reduction in CFU, so I didn't show that, but they did examine microbiology.
But does that not suggest that Pseudomonas therefore is the main problem? Like with everybody kind of working with microbiomes and various things like that. I mean, actually a Pseudomonas phage is what kind of saved her. I'm just wondering kind of where the what the thinking on that is.
I think if you have a clinically relevant pathogen in your lungs, you want to treat that, but that doesn't mean that the microbiome is not also driving other dynamics, right? It's like saying my house is on fire, I should put that out, but also I'm not going to buy a roof because the house is on fire. Like there are other things going on, right?
Oh yeah, I mean, I'm just thinking. They could be driving the Pseudomonas dynamics, but I mean, in that one patient. With Pseudomonas. It doesn't mean to say in the next patient it's going to be Pseudomonas, but then you've got a problem like with phage that, oh God, our Pseudomonas phages don't work against this person, so you've got to also have a bunch of phages against other bugs as well.
People are looking at other bugs, right? So the nice thing about phages is you can find them pretty easily. There are several initiatives looking to try and set up, banks of phage to be used as clinical therapies. They seem to be doing reasonably well. They've got some outside funding now to actually help with that. But yeah, I mean, ideally, I think maybe I didn't stress this enough. Phage therapy is a addition to conventional therapeutics. Antibiotics work too well to sort of think, okay, we're just going to use phage therapy. We should be working in ways we can use phage to save antibiotics for really serious cases.
Hey James, can I ask a question?
Sure, of course.
So, I mean, the coevolutionary dynamics, I mean, like you said, lots of people are doing this, and I think you think about it, right? I mean, it's really clean in vitro, and that's kind of what evolutionary biologists like. They like clean stuff, right?
Yeah.
And so, but it's not going to be clean in the patient. And in fact, you may not even be looking at, other than antibiotics, I mean, the biggest killing factor in the human is the host, right? Which has a whole plethora of ways to kill that have all these different mechanisms. And you also have this sort of, you worry about, I guess, when I think about this, I think about all of the horizontal gene transfer that goes on. We know it happens, right, from these bugs, right? You start out with this really diverse population you can share, and all of these are going to have huge impacts, right, on these dynamics. How can you get to that from your system?
So just on the same page. I mean, how can I get to understand what's being driven by the
How
phage
can you
killing?
understand anything in the human with what you're doing now? Like, just give, you know what I mean? Like, I like the experiments because they're clean, right? So you can get something out of it because it's clean, right? And you can, but the evolutionary biologist has been doing this with microbes for, 15 years or something. Take something, some theory, and you go into the lab, you set up something and you test it and you can show it. But when you go out in nature, it's, particularly in the CF lung, it's pretty dirty, right? It's just a really dirty system. And I just wonder.
Well, so I think we've got an ideal system here to test exactly that question, right? So we know we have patients who are still producing Pseudomonas. So my question being, coevolution. We know we have patients who are still, throughout the entire time they were given phage therapy, we can still test, they still produce Pseudomonas colonies. So we can go in and we can ask exactly how resistant those colonies are on average to the phage therapy, and also capture the phage at that same time point. So I fully agree. I may find actually the system is too dirty to test, but I can't think of a better in vivo setting to actually be able to try and answer those questions. And we can ask about how useful evolution theory is here, and when the big picture, again, I think maybe I didn't sell it enough, but when I look at this patient's data, I'm like, why aren't we doing this for more CF patients? This woman's alive because of phage therapy. I love the idea of studying the coevolution dynamics, but the principal idea should be we should be giving this to more people once we understand it better.
James?
It could be you're not doing it just because she's the only case where it would really work. Because
we know the
perfect bug at the perfect time, at exactly the time there was nothing else to do. And I know it's been done on a few other people, but even if it's been on 10, I mean, and there's 30,000 of them, it's still not many, right? So I guess that's what I'm trying to get at, with this is,
Paul's group are doing clinical trials now, so hopefully, they're expanding the number of people they're going to use.
Yeah, this is Mark Hay. Can I jump in for a second as an outsider?
Sure.
So I think what Marvin's sort of asking is some of what questions I had, but I also, while you were talking, looked up. You said you're interviewing at University of Tennessee. Is that correct?
Mm-hmm. That's okay. Idea, yeah.
So their department's very like ours. It has a group of ecology evolution types. It has cell molecular, micro, etc. And to answer that question, I would have simply said, look, we've got one patient that we've run this on, but I just told you we have 8 more that I'm now funded to start looking at. So I realize that a sample size of 1 doesn't answer it, but we're scaling that up, and that's our next thing to do, And lots of times, if you have a powerful enough approach, the fact that it's really dirty doesn't matter. You've got something that explains 40 to 70% of the variance, and that's great. But just a few things in general for talk. Do you know, is like the microbial group going to decide who gets the job, or is it a departmental-wide decision?
I'm not sure.
Yeah, you almost never are. And even if you're sure, you're probably wrong. I
will say I looked up the same thing, and if let's say I go there, then I would be more on the side of the microbiology. Consider, everyone else again, as you said, it's more ecological. I have a toe in ecology, but I am trained as a classic microbiologist. So I believe that's what they're looking for.
Yeah, and so one of a few things that I would suggest in the job talk is just spend a little more time right at the first, sort of explaining about Pseudomonas and CF and that stuff. There was a bit of that, but some of it passed over because some of your ecologists won't know what causes CF and won't even worry about it. Okay.
I always have more time at the job talk. I have a full hour, so I try to keep this
sure.
30 minutes.
I mean, this is only a sentence or two, just saying a lot of people suffer from this. This is a major agent we think's caused it. I'm working on the following. And then there was a place in there too where you said, one, I would suggest putting some stats on your data slides. What's significant, what's not, what the sample size is. If you can, because the ecologists will appreciate that. And then there was a place where you said something was not significant, but it was a bit higher. For the ecologists, if it's not significant, it's not higher. You don't know.
So it's it was done. That was done with some Bayesian stuff. It was the effect size is bigger. But again, I didn't want to get into the weeds of that, but I get your point. Yeah.
Don't get in the weeds too much. But there's also I know for some biomedical treatments and stuff, people are getting medications that have an effective. The p-value on those things is something that an ecologist would just throw out and never look at. It's like 0.6, and the argument is, well, there's a 40% chance it's actually helping. And so either explain that or go on. But I wouldn't give it for the ecologist, but I'd throw in these little extra things because often when we hear sort of microbial or biomolecular or cellular molecular or sometimes biogeochemical things, there's no stats, and it's kind of nice to see rigor. And you're working on fundamental ecological evolutionary sort of processes. It's kind of cool. So nothing other than that. I thought it was good. I enjoyed it.
Well, thank you, Buck. Thank you so much.
Yeah. I did have one other question, though. I'll quit talking in a second here. But so if you had, in other words, I'm assuming there's a trade-off between evolving resistance to the phage and evolving resistance to the antibiotics, and that you're kind of driving it one way and then hitting it from the other side. One, is that the assumption?
Yes.
Okay, and then two, what if you have antibiotics that work by fundamentally different mechanisms? Can you do the same thing?
So you mean antibiotics resistance mechanisms, or
no, antibiotics that attack the microbe by fundamentally different.
I see. Yeah. So the idea of collateral sensitivity, which has been explored. So people are looking at this idea of you have antibiotic A, which developing resistance to A induces sensitivity to B. People are doing that work. It's some excellent work on that topic. And the idea here, there's even parallels with people working on cancer adaptive therapies. How can you push adaptive therapy to drive yourself this way? It's the same idea. Okay. I think phage has a benefit because of the coevolution, but in principle it's the same. It's a trade-off for A or B.
Okay. And in your experiments, you did not see a difference over the time course of things in the sensitivity when you had phage alone or phage plus the previously ineffective antibiotic. Is that true?
Sensitivity to between the phage antibiotic level and the phage alone level?
I'm sorry. Over time, so you have a patient or a petri dish, either one, that you're applying only phage to or phage plus the antibiotic to. It looked like in some of the data that despite the fact that the antibiotic was there, the microbes still became very susceptible to the antibiotic later on. Is that correct?
Apart from the erythromycin, yes. Everything else reduced in MIC. Erythromycin maintained it at the control level.
Okay. Okay, I'll shut up now. Thank you, though, but I enjoyed the talk.
I appreciate all the help. That was very, very, very great. Thank you.
Hey, James, I have a few questions.
Sure.
For the patient who got the treatment, have they tested if she was more resistant to the phage at the end of her treatment? And
no, they haven't tested it. That's exactly what I'm testing.
Okay, and so have you gotten like any later isolates to see if like even if she was resistant, like maybe the resistance went down over time?
Not for her, but there's potentially for other people. We also have a patient who they have a patient who was went 6 months and then was given the phage again. So there's this exact experiment I'm talking about reapplying later has now been done. Treatment has been done. The experiment the thing I was saying we could do, they've actually done. So we're hopefully going to get their samples to at some point.
So then my next question is something along like Marvin was suggesting. So right now you're going like you have a competing evolution between antibiotics and the phage, but have you considered or tried to look for a phage that would bind to something that is essential for combating like neutrophils? Because like of course you would have to add the antibiotics back in to get phage resistant or susceptible, but like for neutrophils they're always there. So
yeah. I've toyed with that idea. The reason for working the MK01 phage is because we have it, we know it works. But for example, I think my very first attempt at the CF fellowship, I had this idea of what about LPS modifying phage? Will that reduce inflammation and change how neutrophil recruitment happens inside the CF lung? I think there's legs to that idea, but without a phage on hand and without the money to go and start playing with that system, it was dead on arrival unless without funding, I don't think I could start working on that. But yeah, the idea of if you modify the LPS, I think Marvin would disagree with this idea, but if you modify the LPS, could you reduce the host inflammation to the bacteria and maybe reduce how much inflammation occurs and how much damage that does to the cell? But I mean, I can hear Marvin in the back of my head. The LPS is such it's such a nasty target that the effect size you have would be so small, even if you could do it.
Yeah. Is there not like databases or like panels of phage for Pseudomonas? Because for Mycobacterium, there's a huge database.
Not really, no. What we used to have was like the typing phage, but for driving, like the thing I mentioned, how you drive infection, it's just dispersed throughout the literature. And when I say that, I mean literally like from the beginning. Durrell, the second person to discover phage, noted like in 1920 that there are anti-virulence phage. So this is an old idea, but no one, as far as I'm aware, has said here's a database of phage and what the targets are and how they work. That's one. It's hard to do because there's probably co-receptors. There just isn't the impetus to do it yet.
I guess you're not lucky. They're doing that for Mycobacterium, so
yeah. No, that's great, right? Yeah.
James, can I just jump in if nobody else has?
You can do. I can hear you from the room though.
Yeah, but okay. In terms of like following up on what Marvin was suggesting and questioning, and also Brittany, is I think it's important to at some point in your talk to acknowledge your model system is just a simplified system, and kind of like people are doing the treatment, but the detail of how the system works is not known, so you're starting it. So you can kind of like address that question. Yes, we know this is happening, but this is the start. Maybe next step is like include like in your model system, include host factors or, to make it messier to see what is the system or what are the details. It's just a suggestion.
I agree. And as you well know, the follow-on grants I'm hoping to write include more in vivo work and manipulating that environment to make it more realistic.
James, I had a quick question. So you had mentioned the similarity of this kind of trade-off to the collateral sensitivity work that's been done in antibiotics. I was just curious if you mentioned one difference between this and the collateral sensitivity work is this kind of, possible coevolution effect. I was just curious if, the general things that people are starting to see with the collateral sensitivity, namely, trying to reduce the possibility of mutants that are resistant to both and these kind of things, or mutations that happen to disentangle the trade-off or that decrease the trade-off, whether you think some of those same phenomena are going to be the same in phages, or if you think they're different.
I think there's going to be higher order mutations that sort of confer resistance to at different points. That would be my guess. If I had to bet on how resistance will emerge and get around this, there'll be some mutation which moves the bacteria out of I had to catch 22 and actually finds a novel pathway. If that happens inside a stressed environment like the CF lung, I don't know. It's actually quite hard to find phage resistance mutations from active infections because the bacteria, the standard variation gets made so small, and most might survive because of hiding more than natural selection. I don't think that's the case in the CF patients because we found increased sensitivity. If that was the case, I'd posit we'd find the similar levels of resistance, just lower CFUs. So I think we've seen selection here. But yeah, I think maybe those sort of trade-offs might be too costly to maintain inside an infection. I don't know. I think we'll find similar things.
And just with the sensitivity work, there's been kind of an ongoing discussion about dual use at the same time versus cycling and these kind of things. I mean, are these still the same kinds of things that you're going to...
I think the cycling... there's some very interesting stuff in there, right? There's some cool mathematical ideas. I'm not sure the practicality. For example, this at the moment in time, patients who get phage will be on death's door, and we'll just give them the phage and continue giving the antibiotics. So I don't think the cycling stuff is necessarily going to see use anytime soon. But I think there's an argument to say, okay, well, we take our foot off this accelerator, we put our foot on this accelerator, and sort of try find. It's closer to the adaptive therapy side of things, like they're doing with cancer. Where do you find this equilibrium? For most people with CF or most people with these sort of infections, I don't think we're going to have a good system to test that for a long time.
All right. If there are no other questions, thanks everybody, and thanks James again for your talk. Just a reminder, there's still one more spot open to present for this semester. So if you wanted to present this semester, go ahead and head over to the Google Doc and snag that last spot. All right, thanks everybody, and we'll see you next week.
Thank you, everyone.
Bye.